Hello and welcome to this Introduction to Microbiology video. I'm Dr. Ali Roberts, and in this video we're going to look at what a virus is, along with some of the basic structures and symmetries associated with them. And so one of the first questions we must ask ourselves is, what is a virus? And so they are generally defined as being small, infectious, obligate intracellular parasites. This essentially means they are unable to function or replicate without the use of susceptible hosts. And because of the parasitic nature between the virus and the host cell, the host is exploited to produce progeny virions and continue the infection cycle. And so based on how I have described them so far, viruses naturally differ from both our eukaryotic and prokaryotic cells, which are classed as living cells, and therefore they are able to extract and utilize nutrients from the surroundings, growing and replicating independently. Now this essentially means our viruses are classed as non-living, and therefore they must use the machinery of a host cell to replicate, and this is key. Hence the use of the term obligate in obligate intracellular parasite. Now the main differences between our living eukaryotic and prokaryotic cells and our non-living viruses fall into four broad categories: their function, genome structure, growth characteristics, and overall structure. So viruses can also be described as infectious biological entities, which again means they require a host cell in order to carry out its biological function, whereas our eukaryotic and prokaryotic cells are capable of autonomous growth, reproduction, and energy production. The genome of viruses are typically much smaller, often consisting of only tens of genes, and can be composed of either DNA or RNA in a single or double-stranded configuration. And if we were to contrast this to our eukaryotic and prokaryotic cells, they typically rely on a double-stranded DNA genome that is much larger, encoding thousands of genes. Next we have growth, and so our viruses are not duplicated via binary fission as we see in prokaryotic cells, or mitosis or meiosis as we see in our eukaryotic cells. Instead, they use the host's replicationary machinery to assemble and manufacture multiple new copies of themselves within the host cell, making them obligate intracellular replicators. And finally, viruses, unlike prokaryotic and eukaryotic cells, don't have proper cellular organization. They're what we call acellular, lacking some of the basic components of cells such as membranes, organelles, and ribosomes. Essentially, in their most basic form, they are a set of genetic instructions wrapped in a protein coat. And typically, when we look at viruses, they have one of two main structure types. Viruses can be classed as either naked viruses or enveloped viruses. Now the naked viruses are a relatively uncomplex variation and is usually associated with infections of prokaryotic cells, whereas enveloped viruses are much more complex in terms of their structure and genome and are usually more commonly associated with eukaryotic cell infection. Now if we start looking at these structures in a little more detail, we can see key differences in complexity between the two. So here we have our naked virus, which is composed of a capsid, which is a special protein coat made up of repeating subunits known as capsomeres. And within the capsid you will find the genetic material of the virus and possibly some viral proteins. Now together these components make the nucleocapsid, and essentially it allows the genetic material to be protected from the harsh external environment whilst also allowing the virus to interact with unique structures on target cells. And if we now compare this to our envelope viruses, we can see unsurprisingly that the outer part of the virus consists of an envelope, and to be more specific, this envelope is formed from the cytoplasmic membrane of the cell that produced it, which has various viral proteins associated with the surface. And if we were to look inside the envelope, we would find the capsid proteins and genetic material that make up the nucleocapsid, as we saw for our naked viruses. Now in our enveloped viruses, the function of the envelope as the outermost layer is to protect the virus and allow it to interact with unique structures on target cells. However, in order to achieve this, it must remain moist so that it has ample protection within the environment. Okay, so one of the interesting things about the nucleocapsid of viruses is that as the capsimeres assemble, they follow two main basic patterns of symmetry. The protein subunits can arrange into a helical or an icosahedral shape. And in some instances, as we can see in our T4 bacteriophage example on the right, additional proteins can associate with the main structure to develop complex three-dimensional symmetries. Now we know these different symmetries exist because through our development of electron microscopy, we can visualize them with different electron microscopes. And so on the left, we can see the helical nucleocapsid of the tobacco mosaic virus, which causes tobacco mosaic disease. Then in the middle, we have the icosahedral nucleocapsid of the adenovirus, which is responsible for respiratory infections and cold-like symptoms. And finally, on the right, we have the complex symmetry of the T4 bacteriophage. Now, this is a special type of virus because, as its bacteriophage name implies, it is only capable of infecting bacterial cells using the spider-like appendages at the bottom, which help it to anchor onto the surface of very specific bacterial cells. Now, one final and important thing to remember when we're describing the symmetry of our viruses, such as the helical symmetry in the instance of the tobacco mosaic virus, is that it refers to the shape of the nucleocapsid, not the shape of our virus, meaning when it comes to the overall shape, viruses can look very different. And so if we were to look at both the influenza virus and coronavirus, both have a helical nucleocapsid just like the tobacco mosaic virus, but they do not appear helical as they are enveloped viruses. So if we were to look at them with an electron microscope, the helical nucleocapsid would be contained within the envelope and therefore they look different. And that essentially wraps up the basics of viral structures. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.